Hello everyone, this is Vidya Patil working as a postdoctoral fellow in Department of Botany, Savitribai Phule, Pune University. Today we are going to see Plasmodesmata. Plasmodesmata are small channels which connects to the neighboring cells and passes small molecule from one cell to the another cell. Plasmodesmata are present in all plant groups from lower to the higher plant groups. Plasmodesmata helps in cell-to-cell communication and translocation of solids. So, in this module, we are going to see the following objectives. Introduction to Plasmodesmata, structure of Plasmodesmata, functions of Plasmodesmata. In this slide, we are seeing Plasmodesmata intercellular junction, where we can see smooth endoplasmic reticulum, middle lamella, primary cell wall, plasma membrane, cross-linking glycan, pectin, cellulose microfibrils, cell wall of adjacent plant cells. And we can see desmotubules in between the cell walls. So, this is the structure of Plasmodesmata. We can see the electron microscopic pictures of Plasmodesmata. Electron microscopy has been an essential tool for discerning plasmodesmatal structure, but one should be aware that the dynamic of plasmodesmatal transport should not and cannot be absolutely inferred from the static images, but these images are still the major source of information on the structure of Plasmodesmata. Here are some electron micrographs. The cell membrane bound structure of Plasmodesmata shows continuity of appraised endoplasmic reticulum, that is ER, in Plasmodesmata, figure A, with presence of electron dense structure known as central rod surrounded by the proteinaceous structure called as desmotubules. Plasmodesmatal cavity is broad at the center and narrow at ends, forming neck and collar structures as seen in figure B. And a key component of collar is proposed to be callose, located in the near wall. There is also presence of cytoplasmic sleeve on the inner side of plasmodesmatal cavity, composed of particulate proteinaceous matter from which spoke-like extensions are seen protruding in the cavity. In 1990, Roberts modified the structural model where he indicated that presence of contractile proteins surrounding neck region. Also, it was proposed that the central rod is surrounded by electron dense strands made up of actin and myosin. Of this, myosin has been shown to have a high activity of ATPase domain, which is implemented in active transport across the Plasmodesmata. In this slide, we are going to see formation of primary and secondary Plasmodesmata in conjunction with PD occlusions, and degradation allows the plant to adjust the extent of symplasmic or supraocular pathways interconnecting cells of a tissue. In the biogenesis of Plasmodesmata, two types of Plasmodesmata are defined by their origin as primary and secondary Plasmodesmata. Primary Plasmodesmata are formed at cytokinesis at the end of cell division in the developing phragmoplast. During development, these simple structures are replaced by complex arrangement. Formation of primary and secondary plasmodesmata in conjunction with plasmodesmatal occlusion and degradation allows the plant to adjust the extent of the symplasmic or supracellular pathway interconnecting the cell of a tissue. The insertion of a second desmotubules into an elongated plasmodesmatal pore can create two desmotubules within a shared pore orifice. With increasing wall extension, the new plasmodesmata becomes separated from the original by increasing deposition of new cell wall microfibrils between the two pores. Central cavities may or may not form between the two pores. Depending on the plant species in question, branch plasmodesmata formation may result from either the fission model or de novo pore formation. Twin pore may or may not remain connected by endoplasmic reticulum in the middle lamellar region of wall. Central cavities develop between interconnected plasmodesmata as a new pore are being formed. Structure and origin of plasmodesmata. A. The structure of plasmodesmata ranges from simple to several different complex forms including branch, H-shaped and twins. And in the figure B, it is showing two types of plasmodesmata are defined by their origin, primary and secondary. Now, we are going to see that first of all the simple stage of plasmodesmata where there is no desmotubules and no endoplasmic reticular correction is found. In the second step, loosely bound strands of endoplasmic reticulum seen during the late stage of the cell plate formation, mostly in the symplastic transport. In the later stage, the strong constriction tightly bound desmotubules are seen. After that, it forms secondary structure, and in the secondary development, mostly it start with the origin from the simple structure, where the middle nodal role may be in controlling the translocation flux. For example, in phloem loading of sugars in sieve elements from the companion cells. Afterward, it forms branch plasmodesmata structure in which desmotubules are branched, and therefore they forms multiple connections. Primary plasmodesmata form between daughter cells as growing cell plate constrict strands of endoplasmic reticulum running through it, and secondary structure development through the pre-existing wall as well. Plasmodesmata are removed after they are formed. Example, the guard cells from epidermal cells, partly due to degradation of proteins. Role of plasmodesmata. There are two main roles of plasmodesmata, that is symplastic transport occurs from cell to cell as seen in figure A, and short distance transport, that is translocation. Example, sugar loading from phloem cells to companion cell as seen in figure B below. Plasmodesmata mostly take parts in passive transport. There are three main type of transport: symplastic transport, short distance transport and viral transport. These are all passive transport. First, symplastic transport. Symplastic transport are the transport where the solute can pass through one cell to other cell, and this is a passive type of transport. In this, possible pathways for cell to cell water or solid movement. Desmotubules is essentially a solid chain of lipid. The central rod is composed of the lipid polar group and a few proteins which can physically occupy inner core of tightly formed lipid bilayer. Cytoplasmic annules or sleeves is occupied by particulate proteinaceous matter which one associated with both the outer surface of the plasma membrane. In cross sections, 7 to 9 gap occur between the particles. In central cavity, spoke-like structures radiate between the plasma membrane and desmotubules. Wide range of plasmodesmatal morphological have been observed and different in length bearing the size of central cavity. Okay, this helps in mostly the transformation of water and solid from one cell to another cell through the plasmodesmata. Viral transport across plasmodesmata. Transport of proteins and viral RNA, that is vRNA, through an individual plasmodesmata between plant cells is depicted in figure. Plasmodesmata are intercellular organelles in plants that allow the passage of molecules between plant cells. Movement through plasmodesmata may allow transcription factors expressed in one cell to move into adjacent cell, thereby regulating gene expression non-cell autonomously. The transcription factor to be transported first interact with a chaperone, that is movement protein, that binds to the plasmodesmatal PD receptor, allowing the PD cell, that is size exclusion limit apparatus to open. The chaperone and transcription factor then move through the pore along the endoplasmic reticulum membrane ER. The chaperone and transcription factor then move through the pore along the endoplasmic reticular membrane ER that traverses the pore. Once in the target cell, the transcription factor is released. It translocates to the nucleus and regulates gene expression. Viral encoded movement proteins, that is MPs, facilitate these steps. Most MPs are multifunctional. First, MPs are required for movement. Second, MPs bind to virus genome. Third, MPs interact with plant cytoskeleton. 4. MPs localize to plasmodesmata. 5. MPs get plasmodesmata. Viral movement proteins interact with following host proteins to accomplish their functions: cytoskeleton, second protein kinase, third chaperones, fourth docking proteins. This translocation takes place in short distance, mostly for the short distance to the neighboring cell. The good example of this is sugar loading. Plasmodesmata plays important role in sugar loading in phloem cell from companion cells. Companion cells all adjoining the sieve tubes elements show the presence of large number of plasmodesmata, and this transport is take place with the help of plasmodesmata. In this, the quantitative micro auto loading graphic techniques show high flux rates for transfer of sugar at sieve element companion cell interface. Selective loading help by the specific carriers, active accumulators of sucrose against concentration gradient requires ATP. Cytochemical studies indicate that the presence of H plus ATPase helps ATPase. Therefore, the gradient of protoplast 0.3% of the ATP delivered from the phytosine which is needed for loading, and in this sugar loading and unloading is take place with the help of plasmodesmata. So, plasmodesmata plays important role in sugar loading in phloem cells from companion cells. Companion cells walls adjoining the sieve tubes element show pressure of a large number of plasmodesmata. Transport is shown to be ATP dependent. So, this is about the sugar loading. Now, we are going to see about the viral transport across the plasmodesmata. Active transport involves intercellular movement of plant viruses. Studies indicate that viruses manipulate the plasmodesmata for transport to different cells. The major mechanism of this type of viral transport has been deduced through studies of the TMV in tobacco, which encodes for movement protein, that is MP, that mediates the transport of non-viron form of the virus between cells. An experiment based on the work of Session et al. That validates the movement of transcription factor from the site of synthesis into adjacent cells is depicted. The transcription factor LIFY, that is LFY, is synthesized only in layer 1 of a genetically altered meristem and then moves through plasmodesmata into the underlying layers L2 and L3 to influence gene transcription of the meristem. That LFY is a functional after moving through plasmodesmata is demonstrated by the activation of a reporter gene that encodes an enzyme beta gluconidase. When X-gluconidase X-gluco is added to the plant, a blue color appears in the cell that express the reporter gene. Plasmodesmata have also been shown to play a role in numerous other plant processes. For example, it has been suggested that plasmodesmata transport gibberellic acid in algae. Electric coupling between cell has been associated with this density of plasmodesmata between the cell, and electrical signals play a large role in the initiation of wound response in plants. Plasmodesmata have been shown to be sensitive to various levels of calcium ions, pH and light, which concludes that the mechanism of plasmodesmata transport are more complex. Plasmodesmata tend to close when there is significant pressure differential between adjacent cells. Again, speaking to a role of plasmodesmata in wound response upon a loss of turgor pressure. The permanent opening or closing of plasmodesmata serve to signify symplastic domains in plants, often compartmentalization various plant function, a short division of labor, such as in guard cells or in the sieve elements companion cell complex in the stem. Plasmodesmata serves as a director of plant growth and development and may help to determine a program of cell differentiation, such as sealing of root and stem epidermal cells from the rest of plant. As shown in maize, a transportation factor Kn, that is noted 1, was transported to all cell layer of meristem except outermost layer, but the Kn1 protein was found in all layers. Thus, it is observed that Plasmodesmata functions are much more complex and it plays a crucial role in transporting materials and signaling molecules intercellularly in higher plants. To summarize this model, we have studied structure and biogenesis of Plasmodesmata, functions of Plasmodesmata in various transports like sympathetic transport, short distance transport and viral transport across the Plasmodesmata of plant cells. Thank you.